Hello dear students, welcome to Bio Prime Points. In today's class, we are going to study a session on the biotechnology part 1, that is principles and processes. This chapter is divided into two sessions, part 1 and part 2. In this video, we are going to discuss the part 1 of biotechnology, principles and processes. Okay, first prime point is the definition of biotechnology. Biotechnology refers to the technique of using live organisms or the enzymes from living organisms to produce products and processes useful to man. So this is the simple and direct definition of biotechnology. It is the technique of using living organisms or enzymes from living organisms for producing useful products or useful services. Biotechnology can be defined as the use of microorganisms, plant or animal cells, or their components to generate products and services useful to human beings. This is an alternate definition to biotechnology. Then according to EFB, European Federation of Biotechnology, the definition is biotechnology as the integration of natural science and organisms, cells, parts thereof, and molecular analogues for products and services. These three definitions are referring to only one section, that is using of other living organisms for useful for the production of products and also for the production of some services beneficial to mankind. That is what is called biotechnology. Then, the principles of biotechnology, the technique of altering the nature of genetic material and or the introduction of it into another organism or another host organism to change its phenotype. You know that every aspect of living organisms is based on the type of genetic material. Suppose we are capable of changing the genetic material in some or another manner, the phenotype of the organism can also be changed. When we are introducing this changed DNA into a new organism, that is a host organism, its phenotype can also be changed. The techniques to facilitate the growth and multiplication of only the desired microbes or cells in large number under sterile conditions for the manufacture of biotechnological products. So these are the principles of biotechnology. So in this session, we have to remember what is actually biotechnology and what are the principles. Okay. The second prime point is the technique of biotechnology. First one, a creation of recombinant DNA. So what is meant by recombinant DNA? A recombinant DNA is a DNA produced by combining DNA fragments of two different organisms. One may be a prokaryote or one may be a eukaryote, one may be a plant or one may be an animal. When the DNA fragments of two different organisms are interconnected, the new DNA produced, the hybrid DNA that's produced is called recombinant DNA. The first recombinant DNA was constructed by Stanley Cohen and Herbert Boyer in the year 1972. They cut the piece of DNA from a plasmid carrying antibiotic resistant gene in the bacterium Salmonella typhimurium and linked to the plasmid of E. coli, Escherichia coli. That means DNA from Salmonella typhimurium is attached to the DNA of Escherichia coli. So both of these are bacteria, but the DNA from two different strains of bacteria are connected together, forming the first recombinant DNA. And this was created by Stanley Cohen and Herbert Boyer in the year 1972. Then the second technique is a gene cloning. Cloning means the production of multiple copies. This recombinant DNA was introduced in E. coli and made to multiply. That means gene cloning, making a number of replica. The replica production is called cloning. Then gene transfer. The cloned recombinant DNA was then introduced into host cells. That means the recombinant DNA, the newly created recombinant DNA is introduced into a new organism. That organism is called a host organism. This is called gene transfer and cultured in bioreactors so as to produce recombinant protein. The protein produced from the recombinant DNA is called recombinant protein. That is called a desired products. So first of all, three mechanisms are there. First of all, recombinant DNA is created. Then the recombinant DNA is multiplied. Then the third one, the recombinant DNA is introduced into a host organism to create the product of our choice. Then, what are the tools of recombinant DNA technology? The first one, cell culture with the desired DNA, restriction endonucleases, these are enzymes, DNA polymerases enzymes, DNA ligases, cloning vectors, and competent host organism or cells. These are the requirements. Okay. The next prime point is nucleases. What are nucleases? Nucleases are enzymes which are capable of breaking nucleic acids, that is DNA and RNA. Nucleic acid digesting enzymes are called nucleases. Nucleic acid digesting enzymes are called nucleases. There are two categories of nucleases, exonucleases and endonucleases. Suppose this is a nucleic acid. It's a nucleic acid chain. And when we are taking sections from the periphery, when we are cutting the DNA from the periphery, it is called exonucleases. When we are making internal cuts, that is called endonucleases. So, exonucleases which remove nucleotides from the ends of DNA, endonucleases which cut the DNA at specific positions anywhere in its length, that means within the length. So, two types of nuclease enzymes are there, exonucleases and endonucleases. In biotechnology, we are making use of a special type of enzyme. That enzyme is called restriction endonuclease. In short, it is called RE. RE means restriction endonuclease. So, as the name implies, it is an endonuclease. That means it can cut anywhere within the DNA fragment. But restriction endonuclease means it will cut only at a specific site. A site is called restriction site or recognition site. Endonuclease which cut the DNA only at specific sites is called restriction endonucleases. The site at which RE cut the DNA is called recognition site or restriction site. The recognition site will usually be a palindrome. Palindrome means the DNA sequences are reading the same forward and backward, which read the same in both directions. For example, GAA TTC. So this is read in 5-3- direction. Okay. So the opposite strand, it will also be GAA TTC. Again in the 5-3- direction. So in the first strand it is reading towards the right, the second strand it is reading towards the left. So both are reading same, GAA TTC. Okay, this is called a palindrome. So recognition site will usually be a palindrome, which read the same in both directions. RE will cut the double-stranded DNA such that a single-stranded extension will be formed at the cut ends. So it will be cutting the double-stranded DNA, leaving a single-stranded extension at the cut site, that is called sticky end, which can be connected together by DNA ligase enzyme. These are the properties of restriction endonuclease. The sticky ends of two strands may be connected together because the sticky ends must be complementary. Then how the restriction endonucleases are named? You know that restriction endonucleases are obtained from prokaryotes, that means bacteria. Based on the name of the source organism, RE are named. So here, 5 components are there. The first letter is the first letter of genus of the bacterium. The second letter is the first letter of species of the bacterium. The third letter is the second letter of species of the bacterium. The fourth letter is the first letter of strain name of the bacterium. And the fifth is a number. The number determines the order of isolation. Okay. So this is how the name of RE is noted. For example, EcoR1 is the name of an RE. The first RE isolated from Escherichia coli RY13. So here, how EcoR1 is created? E for Escherichia, the first letter of genus name. Co from coli, the first and the second letters of species. RY13, R, first letter of strain name. And 1 isolated first. The second isolated enzyme may be called EcoR2 or EcoR3, like that. So, in the naming of RE, the first letter of genus, the first two letters of species, the first letter of strain, and then there is a number indicating the order of isolation. These are the naming pattern of RNA. Okay. The next prime point is regarding the palindromic sequence. As we already studied that palindromic sequences are reading same forward and reverse in the opposite strands. So here 5-3- GAATTC. In the opposite strand 5-3- again GAATTC. In both strands we have to read in the same direction 5-3- or 3-5-. So GAATTC, here also GAATTC, that is a palindromic sequence. But the EcoR1 will cut the phosphodiester bond in between the guanine and adenine. That means it will cut here. The second strand also it will cut here. Okay. So the guanine and adenine, the phosphodiester bond in between is broken by the EcoR1. As a result, the sticky end at one fragment is AATT here. AATT, and the sticky end at the other fragment will be its complementary TTA. Okay, so this is how the sticky ends are produced, and this is the nature of action of restriction endonucleases. It will cut the double-stranded DNA the palindromic sequences in such a way that single-stranded extensions are produced at the cut end, and they are called what? Sticky end, and the sticky end will be complementary in nature. Okay. Now we are moving on to the next prime point, the cloning vectors. Cloning vectors are the carrier DNA. Vector means carrier. They are the carrier DNA which can be combined with the gene of interest in order to make the recombinant DNA. The desired DNA can be connected with the vector DNA forming recombinant DNA. Okay. The plasmids and bacteriophages are commonly used vectors. Bacterial artificial chromosome, yeast artificial chromosome, human artificial chromosome. Bacterial artificial chromosome is called BAC, yeast artificial chromosome is called YAC, and human artificial chromosome is called HAC. So plasmids, bacteriophages, and artificial chromosomes, these can be considered as cloning vectors. And what are the properties of a cloning vector? A cloning vector must have a site called ORI. ORI means origin of replication. That means the point of the DNA from where replication begins is called ORI. Then there is another point called selectable marker. The antibiotic resistant genes present in a vector are the commonly used selectable marker. Selectable marker can be used for its presence in a particular solution. Then cloning site or recognition site. Cloning site means the site at which the desired DNA can be incorporated in the vector. That is called a cloning site. So only when the foreign DNA fragment can be attached to the vector, the recombinant DNA can be created. So there is a specific site. There must be a specific site at which the foreign DNA fragment can be integrated, and that site is called a cloning site. Then the vector must be a small sized one. It's a small size of the vector. These are the four main properties of a vector, cloning vector. Which are they? ORI or origin of replication, selectable marker, the cloning site, and the small size. Then in your textbook there is the diagram of a synthetic cloning vector that is called PBR322. This diagram is very important. There is in this diagram there is a site. That site is called ORI site. ORI site is the origin of replication. Then two red colored regions are there. One is called AMBAR, other one is called TETAR. Ampicillin resistance and tetracycline resistance. These are the selectable marker regions. Then the PVU2, SAL1, BAM-H1, HIN-D3, CLA1, EcoR1, PVU1, PST1, all these are the cloning site at which that particular restriction enzyme can be used for combining a foreign fragment. That means the BAM-H1 will cut the DNA, cut the cloning vector at this site. The PVU will be cutting the PBR322 at this site. Okay, the site at which the DNA, the cloning vector is broken by a particular restriction enzyme is designated here. That means in the PBR322, By using this PBR322, we can use different types of restriction enzymes. Okay, this is a synthetic cloning vector. So these are the prime points of the first session of the biotechnology chapter.